Hi, welcome. This is Dr. Hadi and you are watching Medical Globe by Dr. Hadi. Dear students, today many of the students, they have just told me to record a video on this topic, but the good news is that I know you people will be waiting for my lectures. In today's lecture, the title is Concept of Electron Carriers in Biological Oxidation and Structure and Mechanism of the NAD and FAD. Of course, this is another quite complicated topic for the students. Most of the students want to know these two things, NAD, FAD, or electron carriers. So within this video, the good news is that within this video we have 9 questions, the answer of which we will discuss, and I will teach you these 9 questions answers. The first question, you can write it down and you can pause the video and write the questions. 9 questions we have. The first question is oxidation of glucose to CO2 require electron carriers, right? Second question, mechanism of NADH, how NADH acts. We see this NADH in a lot of reactions, right? So today you will learn the exact mechanism of NADH. What is NADH? What is the structure of NADH and how it acts? How it is involved in metabolism. And the third question is NAD positive charge? Is NAD really a positively charged something or something else? So you will also be given the answer of this. The fourth question is the relation between NADH and dehydrogenase enzyme. The fifth one is vitamin niacin is a vitamin. The deficiency of that vitamin along with NADPH relation and a disease, pellagra. These 3 things will also be inshallah discussed in this lecture. The 6th question is role of vitamin in metabolism. The 7th question is the difference between NADP and NADPH. Okay, you will be astonished today that there are a lot of questions. 8th question is Rossmann fold. 9th one is what is the difference between NAD and FAD. Most of students will be searching these questions around the net and YouTube, but today you will get all these answers in this one single lecture. So keep watching from beginning till the end and never to give up, right? So at the end of my lecture I will take this page once again and I will check any question left. If it is, so I will tell you the answer of this. Okay, so let's start here with the first thing here that I'm discuss is that we have glucose and the metabolism of glucose or the oxidation of glucose, the breakdown of glucose in a very simple step is that when glucose is broken down, it is converted into one form, for example, for simplicity, right? And then glucose is the A is converted into B, and finally glucose is converted into carbon dioxide and water. We call this step as a oxidation of glucose, right? I'm not going to write all these steps, the real structures in between this, but I'm just giving you the idea that the oxidation of glucose means glucose is broken down step by step, one structure into another structure, and finally we got carbon dioxide and H2O, right? This is called as oxidation. So what is most important in this reaction is while the oxidation of glucose, there is the transfer of electron. Right? When glucose is oxidized, this transfer of electron is today's most important topic. When electrons are transferred during oxidation of glucose, these electrons can be transferred with the help of something. There must be something that transfer these electrons from one place to another. So this is not the glucose. Yes, we know these electrons belongs to glucose. These electrons are the part of glucose molecules. But the transfer of electron, which is the real part, which is the most important part, is the electron transfer. So what is that thing which transfer electron during a glucose oxidation? Is the electron carriers. The name indicates, very simple name. The name indicates that these are the substance that carry electron from one place to another, and they are called as electron carriers. So electron carriers is just a general name. Is there any special name for these electron carriers? So yeah, I will say yes. These are the coenzyme and protein that act as electron carriers, and they carry electron from one place to another. So many of the students might also be thinking that what is the need of this electron transfer? Actually, when electron transfer takes place, during this electron transfer we got energy. And on this topic I have already recorded a video lecture on this topic, that electron transfer is associated with energy, so we will not discuss this once again. However, most of the newcomer students who are watching this video, I would like to tell them that whenever electron transfer takes place in the living body or a cell, there is association of energy and electron transfer. When electron transfer takes place, there is energy. So let me tell you something more about these electron carriers, which are coenzymes or protein. These coenzyme or proteins will be discussed now, but the most important the most important coenzyme that I would like to discuss, that today's video is about, which we are going to discuss, is that the first coenzyme is the NAD, right? FAD. Okay, I'm sorry, these are not all coenzyme. If I use the word cofactor, this will be more good, right? You can use the word cofactor in place of coenzyme. So NAD, FAD, cytochrome, yes, these are a few names, FMN, and ubiquinone, right? Cytochrome. All these are just few examples of the electron carriers, right? These are few examples. One is iron sulfur protein. Yeah, these are just the names, a few names of the substance that carries electron from one place to another. Among these, the most important one that I'm going to discuss today with you people is the NAD. Complete information: NAD, NADH, NADP, and NADPH. Even a little bit discussion on the FAD also will be there. A 5-minute lecture will be also there on FAD. But what I would like to tell you about all these is some of them. They are usually soluble in the aqueous environment. Inside the cell we have water, and these are soluble in that environment mostly. Some of them, they are not present in the aqueous medium of water, but they are attached with the cell membrane. Just like if we have a cell here, okay. This is the cell, right? And this one is the cell membrane, right? This is the cell membrane. Some of these electron carriers are the part of the cell membrane, right? For example, cytochrome, ubiquinone, these are well-known electron carriers that are attached with the cell membrane, protein especially. And there are some electron carriers which are not attached to the cell membrane, but they are attached to the enzyme. Okay, so Yeah, this is enzyme, right? We have enzyme here. What is enzyme? Some of them, especially like NAD and FAD. NAD here is attached to this enzyme loosely attached. And what we call to such coenzyme which are loosely attached to the enzyme, they are called as coenzyme. Especially they are called as coenzyme. And there are some other electron carriers which we can call them as FAD. FAD is not loosely attached, but it is strongly attached to the enzyme. So those which are strongly attached and does not leave the surface of the enzyme, they are better called as prosthetic group. However, in some books both of these categories are given the name coenzyme, but there must be a difference between a coenzyme and prosthetic group. A prosthetic group is a non-protein part of the enzyme that is attached with the enzyme. So just like FAD, it is strongly attached. And so we will call it as a prosthetic group, right? So this is prosthetic group, right? So it was just a slight introduction about all these things. Now we will discuss NADH structure and mechanism in full detail. I hope everything is there on the board. You can pause the video and you can draw the diagram. But let me tell you how to write the diagram. In the introduction of the NADH, NADH stands for nicotinamide adenine dinucleotide. Nicotinamide adenine dinucleotide. Yes, so it carries a long name. Nicotinamide, this is for N, nicotinamide adenine A, dinucleotide D. So D for dinucleotide. Now why it is called as nicotinamide, why it is called dinucleotide, I will explain that. Well, the first one, nicotinamide, because in the structure we have, in the whole structure of a nicotinamide, NAD, we have a very well known structure. Here in the red, this structure is called as nicotinamide, that came from a vitamin called as niacin. So here is a vitamin called as niacin. This niacin is a source of nicotinic acid. Niacin is a vitamin, right? This is a vitamin. And from this vitamin, we get a compound called as nicotinic acid. But this is not nicotinic acid. If there was an OH group here in place of NH2, we would call it a nicotinic acid. But as there is no OH group in place of NH2, there is amino group NH2, so we will call them as nicotinamide. And why it is called amide? Because whenever NH2 group comes with C double bond O, we call it amide. If just NH2 group is attached without any C double bond O group, we call it amine. So this is nicotinamide. So this structure is called as nicotinamide. That is why in NAD it is called as nicotinamide. And then we have adenine. Adenine, what is adenine? Adenine is a nitrogenous base we have discussed in the lecture of DNA. Whenever you go to the DNA molecule, inside the DNA molecule, there are very small units that are linked together to form a long chain. Smaller units, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, and so many smaller units. These smaller units, when combined, they make a DNA. One of the name of the unit is nucleotide. Nucleotide. And of course, nucleotide is of two types. One is deoxy, one is ribonucleotide. They make RNA and DNA respectively. So forget about these things today now. Nucleotide is the building block of nucleic acid, right? And nucleic acid is of two types, DNA and RNA. So these nucleotides make up the DNA. So here we have a nucleotide as well. Look, there are 3 parts in one nucleotide. Whenever you see one nucleotide, 3 parts. Part number 1, there must be a nitrogenous base. Part number 2, there must be a sugar. Part number 3, there must be a phosphate group. So these 3 groups are collectively called a single nucleotide. So I know I can't describe the structure of DNA in this lecture. I have recorded this lecture in Urdu medium. In future, inshallah, we will record this in English medium as well. So one nucleotide which consists of 3 parts, you can see this one is the first part, and we call it adenine. That ring that you can see, that is the adenine, nitrogenous base, a nitrogenous base. And why it is called nitrogenous base? Because in this ring we can see there are nitrogen atoms attached here and there, right? Okay, there are double bonds as well, right? You can see them, right? This is just a structure where the nitrogen are very important, right? Otherwise there are many other atoms as well, but there is no need. So this is a nitrogenous base called adenine. Come here, we have a sugar, a glucose, a ribose sugar, a 5 carbon sugar. This one is the carbon number 1. This one is carbon number 2, right? This one is carbon number 3. This is 4, and that one is carbon number 5. So 5 carbon sugar, which is called as a ribose sugar, right? This is the sugar, and here we have 1 phosphate and 2 phosphates. So don't worry about 1 or 2 phosphates. At least now we find that there are 3 components: the nitrogenous base, adenine, sugar, and the phosphate. So 3 group comes. When 3 groups come to us, we call it a nucleotide, and of course it is a nucleotide. So here is the word nucleotide. Next question: why this is called as dinucleotide? It means there must be 2 nucleotide, right? Okay, this is very quite confusing for some students. They are always thinking, why it's called dinucleotide? You are agree with the point that this is 1 nucleotide. Okay, so where is the second nucleotide? This one, this one is the second nucleotide. But the problem is that in the second nucleotide we have sugar, S. Yes, we have sugar. We have phosphate. Okay, we have phosphate, but we do not have a nitrogenous base, just like adenine, guanine, cytosine, thymine, or uracil found in the nucleic acids. In place of the nitrogen, in place of the nucleotides, we have nicotinamide ring. I know this is not a nucleotide, but it looks like the pyrimidine. It looks like pyrimidine. Oh, what is pyrimidine? And don't worry, pyrimidine is a single ring nitrogenous base. As we discussed in the nucleic acid, pyrimidine may be cytosine or thymine. So cytosine and thymine are the single ring pyrimidine bases. So this is because it is single ring and looks like pyrimidine, hence we will consider it that it is a a nucleotide, that it is a nitrogenous base. So if we consider this one is a nitrogenous base, right, of course it is nicotine, amide, sugar and phosphate. So this is another nucleotide. We got one nucleotide and second nucleotide. For this reason we call them dinucleotide, right? And this was all about the structure. And one thing more, the most important structure in this whole diagram is the one in the red color. The one in the red color, which we call it nicotinamide, the whole function of the NAD lies in that structure. So let me tell you something more special about this structure. Okay, you can erase this, right, because it's not important to us now. You can just here you can write here a box, write a box here, and in that box we have adenine, right? In that structure, if you see here, we have a positive charge on the nitrogen. If you remember, maybe some of the students remember that nitrogen has 5 electrons, 3 usually it uses in bond formation, and the 4th and 5th electrons are present in the form of lone pair, the 4th and 5th electrons. So, right, whenever nitrogen gives its lone pair to some other atom for coordinate covalent bond or for covalent bond or for just simply a bond, then what happens? Nitrogen gets a positive charge. If the nitrogen is given the same electrons, then it will loses its charge, it will come back to its original position. Similar is the case with that nitrogen. This nitrogen has given its lone pair of electron in the bond formation. So what will happen, that this structure, it will be given 2 electrons and 2 protons. How? This is not good to say 2 electrons and 2 protons. In reality, this structure will be given a hydride ion. What is hydride ion? Let me tell you. When there is a negative charge on the hydrogen, we call it hydride ion, and when there is positive charge on the hydrogen, it is called as proton. The question is that how it is formed. Look, 2 hydrogen we have, right? This electron belongs to this hydrogen and this electron belongs to this hydrogen, right? Every hydrogen has its 1 electron. If we transfer this electron, give it to this hydrogen, so what will happen? This hydrogen will get 2 electrons and this hydrogen will be without electron. The hydrogen without electron will become H plus proton, and the hydrogen with 1 more extra electron will become hydride ion H minus. Okay, NAD gets electron in the form of hydride ion. Okay, so what we call to a structure that has no electron, that has lost its electron, is called the oxidized form. So now what you can see, this structure is in oxidized form. So this is the oxidized form, because nitrogen N plus has used its electron. By mistake, some of the students think that that NAD plus is positively charged, which is wrong. And maybe this is your question number 3: is NAD plus is positively charged? No. NAD plus is not positively charged. Actually, we write the plus charge on the top of the NAD to show that it needs electron to give it to the nitrogen. It needs electron to reduce. Otherwise, it is in oxidized form. It will get electron to become reduced. But it is not positively charged. Let me tell you the reason. Come see in this whole structure. we have one positive charge on the nitrogen and we have two negative charges. If we have two negative charges on the NAD, why we should call it NAD plus? We should write NAD minus. No, actually this is not the case. This structure is in oxidized form, hence we write a positive charge on it. Okay, what happened? It will get two electrons in the form of hydride ion, where the two electron will be given. If you see here in that whole structure, only red nicotinamide ring has been shown to you, but the remaining whole structure, this whole structure, it is not of our interest. So in place of this whole structure, I'm just going to write R, simple R. R means that this whole structure lies here. So we will not waste our time by writing all these, because the main part is the nicotinamide ring. As I told you, the main part is nicotinamide. Now what will happen, that the two electrons, the hydride ion, they will come here on that carbon, on this carbon, right? So what will happen, this is a pi bond, right? It's a kind of electron. So these electrons will come on that side. It will become like this, right? And this bond, of course, pi electrons, the two electrons, they will be given to the nitrogen atom, and once the two electrons are given to the nitrogen atom, nitrogen will get its lone pair. These two electrons came actually from the pi bond which was present here, right? And what will happen if this pi bond come here? Of course, a positive charge will create, a positive charge will be created on this carbon. And remember, we don't show the carbon atoms. Every corner has carbon atom, right? So on this carbon, a positive charge will be produced up here because that bond will be shifted. So what will come to neutralize that charge? This is the H minus, the hydride ion. As I already told you, we have two hydrogens. From these two hydrogens, we make one hydride ion, and that hydride ion will come on this positively charged carbon, and it will attach here. The charge of positive carbon will be disappear, and now you can see that we have carbon has 4 valencies. And it's okay. The hydrogen in the black color, this hydrogen is actually the hydride ion that came to the nicotinamide ring, and now there is no positive charge on the nitrogen. This is whole structure. We will write this structure as NADH. Yes, it was NAD, right? And now it become NADH. Remember, there are still two negative charges in that one, in the form of the phosphate of the oxygen atoms, right? There is also two negative charges. But we will not show them. The positive charge, the presence of positive charge and the absence of positive charge only reflects electrons. When the electrons were removed from the nitrogen, they were used, then we write it NAD plus, means the oxidized form of NAD. And when the NAD get electron from somewhere, from the substrate, from the glucose, or from anything else, when they get the electrons, then it become NADH. Not only electron, of course, hydrogen also. So this will be called as the reduced form. And this is the H plus, that we write it separately by the side of NADH, H plus. I think you got the idea where the H plus comes. It is actually here, because we have two hydrogen atoms. We had two hydrogen atoms. One hydrogen become H minus and the other become H plus. So this will be the H plus. Most of the time we will write it NADH plus H plus. So in many reactions we may not write the H plus again and again. We will only write the NADH. So this was the structure and the mechanism. Now here I have just mentioned one point for you people, that NAD which is a coenzyme, it is linked to the enzyme called dehydrogenase, and there is a class of enzyme called as oxidoreductase enzyme. Oxidoreductases are those enzymes which are involved in oxidation and reductions. I have also recorded a video lecture on oxidation and reduction. You can watch this video by just typing biological oxidation in the chapter of biological oxidation. You will find. So now we will come to the topic that if the NAD is a coenzyme, there must be an enzyme. There must be an enzyme, and with this enzyme there must be attached NAD plus or NADH. This is the protein part and this is the non-protein part. What is the name of that enzyme that is usually linked with NAD? The name of those enzymes are called as dehydrogenase enzyme. And remember, dear students, you will find that enzyme dehydrogenase a lot of times, so many times, frequently you will find these an enzyme name in the metabolism of carbohydrate, lipids and protein. So now a slight some more points about these enzyme and NAD. At the question number 8 question, what is a Rossmann fold? A Rossmann fold is the place on the enzyme. Let's suppose this is the enzyme, dehydrogenase enzyme. There is a place on the enzyme at which the NAD is attached. This place, this site is called as Rossmann fold, and fold because here protein chains, you will find protein chain, right? So we call it fold. And you know what you will find here in that protein chain? You will find six beta chains of protein, 6 beta chains of protein, and 4 alpha chain. Don't worry about alpha beta, don't worry. The alpha and beta are actually the organization of protein. Proteins can be found in primary structure, secondary, tertiary, and quaternary structure. Among these 4 organizations, the second one is the secondary structure of protein is actually beta and alpha. So don't worry, alpha and beta are actually the secondary structure of protein, and at the Rossmann fold you will find these chains. Total 10 chains, 6 beta and 4 alpha chain. Right. Now one more special point is we already discussed that NAD is the friend of enzyme, a partner of enzyme, but it is loosely attached to the enzyme, means that here I think now we don't need that structure, so I'm going to erase it. There we have an enzyme. This is one enzyme, right? Enzyme E1, and this is another enzyme E2, right? NAD attached to one enzyme, right? Of course it is in oxidized form. So with the help of the first enzyme, it will get its 2 electrons and hydrogen, right? When it gets electrons and hydrogen, of course it will become NADH. Okay, what will happen? As we discussed, it is loosely attached. So what will happen? It will leave the surface of this enzyme and move towards another enzyme E2, and yes, E2 here, and it will come here NADH. Here it will give its electron and hydrogen back to another reaction. So NAD got oxidized got electron in hydrogen, and at other place it will give back its electron in hydrogen to another reaction. So it is reversibly oxidized, right? Reduced and oxidized. This is the characteristic of the electron carriers. Most of the electron carriers, they can be reversibly oxidized and reduced. If it is oxidized at one side, it can be reduced at another side. So NAD can be oxidized and it can be reduced back in oxidized. So one example of this is we have an example of this. The example is glyceraldehyde 3-phosphate. We have a compound glyceraldehyde. Oh my god, sorry, I have to write it here. Glyceraldehyde 3-phosphate. Just let me check whether, yeah, okay. Glyceraldehyde 3-phosphate, it is converted into phosphoglycerate. It's converted into 3-phosphoglycerate. You are not concerned with the structure of these. We are concerned with the concept. There is an enzyme called glyceraldehyde 3-phosphate dehydrogenase. Okay, this is the enzyme. This enzyme will convert glyceraldehyde 3-phosphate into 3-phosphoglycerate. Remember, there is NAD plus in oxidized form. With the help of this enzyme, glyceraldehyde 3-phosphate dehydrogenase, the NAD becomes NADH, right? So this is the task, the duty done on the surface of one enzyme. Now what will happen? Another reaction will start, which is acetaldehyde. So here is another reaction, acetaldehyde, right? Acetaldehyde, this acetaldehyde will be converted into ethanol, and what happened with the help of NADH, the acetaldehyde will convert into ethanol. Looks like acetaldehyde is oxidized and this is the reduced form. So who gave the hydrogen electron? This is the NADH. There is another enzyme. The enzyme is alcohol dehydrogenase. Alcohol dehydrogenase. This is another enzyme. So this is so special. The NAD attached to one enzyme gets reduced, leave the surface of that enzyme, come to another enzyme, attach with that enzyme, give its hydrogen electron back and oxidize. So it is a kind of movement between one enzyme and other enzyme. Okay. Now that is our lecture. Now what left is, okay, what is the main difference between NAD and NADP? Yeah, some students always ask me questions, sir. Sometimes in books we see NADP. Don't worry, if this is the structure of NAD, in NADP you will find NADP. You will the same structure just like NAD. You will find one more extra phosphate group. Okay, there were two phosphate, so it will get a third phosphate. So it is called as NADP. Question is that what is the purpose of these two? If there is NADH and then there is NADP. Actually, there are two types of chemical reaction. One is catabolic, and one is anabolic. Okay, anabolic reactions are those in which substances are combined to make new substance, and catabolic reactions are those reactions where a compound is broken down. So in catabolic reactions you will find NAD, and in anabolic reaction you will find NADP. Of course it is reduced form is NADH and its reduced form is NADPH. So that is this was the difference between the catabolic and NADH and NADP. And there is one thing more, that the ratio, this is also here, it has NADP oxidized form and NADPH the reduced form. It has NAD positive charge oxidized form, NADH the reduced form. Here in catabolic reaction the NAD ratio is high amount as compared to NADH. This oxidized form is much more as compared to the reduced form, but here the reduced form is much more as compared to the oxidized form. So this is one thing that I discussed with you people. Now we have one more question. Okay, the last question is the difference between NAD and FAD. Not a difference, a kind of comparison between NAD and FAD. So if someone asks me or you, what is the difference or what is the comparison between the NAD and FAD, so I will give you the answer. NAD is loosely attached to the enzyme, but FAD is tightly attached to the enzyme. If it is loosely attached, so we call it coenzyme. If it is tightly attached, we call it prosthetic group. Right? So this is called as prosthetic group. NAD require electron and hydrogen in the form of hydride ion, H minus, hydride ion. But FAD require this in the form of hydrogen atom. Both require hydrogen, but this get in the form of hydrogen atom and this get electron hydrogen in the form of hydride ion. So there's a very big difference between these two. Okay, in NAD the main functional part was or is nicotinamide. The main functional part is the nicotinamide. Nicotinamide came from the vitamin niacin. Wow, niacin. And here in FAD the main part is isoalloxazine ring, came from riboflavin vitamin. And you saw the structure of nicotinamide. You can write the structure of isoalloxazine. This is very, very simple. I'll just tell you how to write the structure of isoalloxazine. Here, you can do like this, I think. Yes, you can. I know you can. You can do like this. Okay. Yes, it is too simple for you people to write like this. Write 1, 2, 3, and 4. Right? Locked. This is a structure, a simple structure of isoalloxazine. You will find there are double bond O, you will find it here, and one double bond here, a nitrogen here, right, and another nitrogen here, another nitrogen here, and of course here double bond. So this is the structure of isohexane ring. This is the main part of the FAD. As we have discussed the main part of the NAD, so here will be the main part of the FAD. Okay, so the structure also came here. A comparison was also made between the FAD and NAD. Because of the shortage and lack of time, we will not be able to discuss the FAD, but any student who got the structure and mechanism of NAD, he or she would definitely be able to get the structure and mechanism of the FAD. Okay, one question which was the question number 5. Question number 5 was, is there any relation between the vitamin niacin? Here is the vitamin niacin and NADH and the disease pellagra. Yes, there is a big relation. There was also a question, the role of vitamins in metabolism. Most of the students, they think, what is the relation between vitamin and metabolism? Vitamins, which we know required in very small amount for the body, for normal growth and development of the body. Some of the vitamins are the friends of enzyme, are the partner of enzyme. They help enzyme. Now you think, if a vitamin help enzyme, and enzyme is involved in the metabolism, and metabolism is metabolism provides us growth and energy. Now you think, if there are no vitamins, who will become the friend of enzyme, the partner of enzyme? If the enzyme is without its partner, enzyme will not be able to metabolize, and you will not see a growth or energy in the body. One example of this situation is the absence of one enzyme called niacin. Vitamin niacin. Vitamin niacin give us nicotinamide, right? And that nicotinamide is the part of the dehydrogenase enzyme. Now see, if there is no vitamin niacin, so there is no nicotinamide. No nicotinamide means no NADH. No NADH means that our enzyme dehydrogenase, that enzyme will be not happy. He is no cofactor, he is no partner. So this enzyme will not be able to help metabolize reactions in the body. So a disease will occur if that vitamin is deficient, if that vitamin is absent. The disease name is called as pellagra, and pellagra has its history that in 1912 to 1916, 4 years, 10,000 people died of this disease pellagra, because at that time they were using maize which was deficient in that vitamin, so the people suffered from the disease. And then a scientist came to know that the cause of that disease pellagra is the deficiency of a vitamin called niacin. And what happened in pellagra? In pellagra, we can memorize it with triple D. One D is for diarrhea, other is for dementia, and third one is for dermatitis. Diarrhea, everyone know very well. Dementia, memory loss, and dermatitis, infection of the skin. So these three symptoms belongs to the pellagra disease. So those people who are deficient in that vitamin, these people will suffer from that disease. So if that disease came to a person, so he or she will be given the vitamin niacin in the diet. Okay, I hope you got this lecture. Just as I told you that we had 9 questions, I just want to confirm that mechanism of NADH, okay, NAD positive charge, done. Dehydrogenase enzyme, okay. Vitamin niacin, okay. Role of vitamin in metabolism, okay. Difference between NADP and NADH, Rossmann fold, and what is the difference between NAD and FAD. Thank you students for watching this video. I hope you like that video. If you like it, just have a thumb up, and as usual I say, always keep smiling, and I want a very happy, a very pleasant smile on your face. Remember me in your prayer. Thank you. Bye bye.